Welcome to my channel Learnovate. In this video, we will going to discuss about the introduction to anemia and its classification. So this is the first lecture of pathology blood module in which we will going to discuss the pathology of anemia. But before going to discuss the classes of different anemias and what are the characteristics and pathogenesis of complications of these anemias, first we will going to know the simple concept of anemia. Here I have written that the anemia is defined as decreased hemoglobin, or it's defined as a decreased oxygen carrying capacity of blood. Simply when there is a decrease in the RBC number or when there is a decrease in the hemoglobin concentration, this ultimately will going to decrease the oxygen carrying capacity of the blood. And when there is a less oxygen, there is hypoxia, there is injury to the RBC, this will going to cause the anemia. That is decrease in the hemoglobin concentration and decrease in the iron concentration as well. Because hemoglobin is made up of globin. Separately we have two parts in hemoglobin. The globin, that is the protein, that is a chain of alpha and beta subunit, while the heme part of the hemoglobin, heme part contain the iron. So both of these are important for the synthesis of proper and normal hemoglobin. If any one of this, if the heme part will get decreased, or when there is a deficiency of iron, that's called nutrition deficiencies, and when there is a deficiency of iron, there will be not functioning of heme part and there will be again the concentration of hemoglobin decreased. Or if there is a deficiency or destruction or mutation in the formation of globin chain, or there is a destruction of alpha or beta chain in the disorders that are thalassemia, sickle cell anemia, what happens? There is again the decreased amount of hemoglobin form. So both of these, that is the globin part and the heme part, are equally important for the formation of normal hemoglobin. And what happens when there is a decreased hemoglobin concentration, either due to these both characters, or either there is a decreased amount of RBC, or there is a decrease, you can say there is a different in the shape of RBC, there is increased destruction. This may all causes the anemia, but the journal concept about the anemia is there will be a decreased hemoglobin concentration, oxygen carrying concentration of the blood. Now what happens when there is a decreased hemoglobin or decreased oxygen concentration? There will be an increase in the erythropoietin level as a feedback. What happens? Our brain detect this condition of the hypoxia and anemia. This will going to give the signal to our kidneys to release the erythropoietin. And what is erythropoietin? Erythropoietin is a protein. It is a hormone that is being released, and the erythropoietin has a function in the bone marrow. It will going to release from the kidney. It will go to the bone marrow to increase the erythropoiesis. Okay, so erythropoietin from the kidney, it will going to bone marrow, and where in the bone marrow it will going to increase the erythropoiesis. And what is erythropoiesis? It is simply erythro means RBC and poiesis means formation of RBC. Synthesis of RBC increase from the bone marrow, that is because of the feedback of the erythropoietin. And now when this erythropoiesis is increased in the bone marrow, this is called intramedullary erythropoiesis. Why intramedullary? Because it is inside the medullary cavity of bone marrow, or inside the medullary cavity of long bones. The other erythropoiesis that take place outside the marrow cavity is called extramedullary erythropoiesis, and this happens in many types of anemia in which there is an increased RBC production or erythropoiesis that occur in liver or spleen or in other parts of the body, and this is called extramedullary erythropoiesis, I have written hemopoiesis, it's erythropoiesis. Okay, so extramedullary erythropoiesis occur outside the medullary cavity or in areas of the liver and spleen that are not related to bone marrow. This both type of erythropoiesis will ultimately going to increase the concentration of reticulocytes. Okay, so reticulocytes will going to increase. These reticulocytes, what are reticulocytes? They are the immature RBCs. This immature RBC, when increased, we will going to find reticulocytosis in most of the type of anemia. And the reason behind this increase in the reticulocytes are, I have told you, because of the increased concentration, increased erythropoiesis in the bone marrow and in the extramedullary tissues. So this will going to increase the reticulocytosis, and we will find reticulocytosis as a clinical finding and a lab finding in the certain types of anemia. So this was the basic mechanism of anemia. Now we're going to discuss the classes of anemia in the next part. Now again, in this part of video, we will going to discuss the classification of anemia according to the cause. I have made it simply so that you guys will easily get to remember the journal and the major concept around each anemia and what are the main cause of each anemia. So according to the cause, we have divided the anemia in basically 6 to 7 types, and you can divide these 6 to 7 types into more general types. So this is a basic cause related classification. Starting from the first cause, that is trauma. Trauma, that is the mechanical trauma, the surgical trauma, or any inside your body there is a chronic or the acute disease going on. When there's a chronic disease or there's a mechanical injury, what happens, there is a blood loss. Simply there's a blood loss, there's an excessive bleeding that will going to occur, and this will going to cause the hemorrhagic type of anemia. This hemorrhagic type of anemia is because of blood loss simply, and this is the reasons we have discussed. They are the trauma. Second type of anemia is because of the increased destruction of RBC. Increased destruction of RBC, and this increased destruction of RBC can occur in several conditions. But now what happens, we will going to discuss the increased destruction of RBC will going to cause the hemolytic anemia. Okay, so the point we have to remember is the increased destruction of RBC can cause hemolytic anemia. Now what are the conditions when there is an increased destruction of RBC? There are certain conditions in which the RBC destruction increases, and these conditions occur when there is an abnormal formation of RBC, abnormal shape of RBC. So that's why it cannot go through the spleen, it cannot go to the blood vessel and destruct easily. So these are the conditions, example hydatidic spherocytosis, electrocytosis, and other conditions in the RBC shape has been changed or become abnormal. So there will be a hemolytic anemia seen. The third condition is RBC enzyme deficiency. Any deficiency in the formation of either the globin chain or either the heme part. Okay, so any deficiency related to globin chain, mostly it's related to the heme part, or the deficiency of the enzyme that is important for formation of the normal RBCs are destructed or deficient. There will be formation of an anemia. This is called hereditary anemias. Mostly these hereditary anemias are glucose 6-phosphate dehydrogenase anemia or pyruvate kinase anemias. Now the next is the hemoglobin synthesis disorder. This is the main one, that is in which the globin chain is disrupted or there is an abnormal or mutated globin chain. So we have just discussed that there are two parts of globin chain, the alpha part and the beta part. So when there is a mutation, when there is a destruction, or when there is a decreased synthesis of globin chain, the anemia will be going to occur is hemoglobinopathy. That is, it is maybe a condition of sickle cell anemia, or it may be a thalassemia like disorders, that is alpha or beta thalassemia. This is also a type of anemia. Now the fifth one is impure RBC production. Now there is no destruction, there is no deficiency, there is no abnormality in the RBC, but there is decreased production of RBC. And this impaired RBC production or decreased RBC production is occurring in the conditions called bone marrow aplasia, or this type of anemia is called aplastic anemia, in which there is a decreased production of RBC from the bone marrow. What happens, erythropoiesis is being stopped at the level of bone marrow, either due to any other cause that are not related to this basic video. So this may cause the aplastic anemia. Now the last one is the autoimmunity. Autoimmunity means that our body makes self immunity, like it will going to make the RBCs as foreign, make our self RBCs determined as foreign particles and destroy them. So what happens in this autoimmunity deficiency or in autoimmunity diseases, there will be a formation of immunohemolytic anemia, and this is the worst condition in which your body make immunoglobulins against RBC. Okay, so what happens, there is RBC carrying these antigens, and when the macrophage come, this will going to opsonize this RBC and break up this RBC, and there will be a hemolysis. Okay, so what is hemolysis? Hemolysis is simply the breakdown or destruction of RBC either in the any cause of anemia. So guys, these are the some of the 6-7 major causes of anemia, and what are the anemia's cause I have discussed according to the classification, according to the cause. Now smallest important thing that is being remained, are these reference values that are used to measure the amount of anemia or the you can say the severity of anemia. We check through these important values, and that number one comes is the hemoglobin level, that is normal in the male is about 30 in the male it is about 13.6 to 7.2, in the female about 12 to 15. So it's in the range of 12 to 17 gram per dl. So this is a normal hemoglobin level. If it is decreased, then we consider it as an anemia. Second one is the MCV, that is the mean corpuscular volume, that is a volume of hemoglobin inside the volume of RBC. The normal range of MCV is 76 to 100 femtoliter, and if it is decreased, it may be normocytic, microcytic or macrocytic RBC. Then MCH, that is mean corpuscular hemoglobin concentration, that is about 27 to 33 picogram. Normal. If it is decreased, it is hypochromatic, and if it is increased, it is hyperchromatic. Then MCHC is the mean corpuscular concentration of single RBC in a given volume of packed RBC. So it is expressed in gram per deciliter, that is about 33 to 37 gram per dl, that is a normal value. So these are some reference ranges that you have to remember, and you have to learn it by heart, so they are being diagnostic feature for any type of anemia. So guys, this was all about the anemia. I hope it will be explained well. Thank you for watching. Subscribe for more, and we will going to discuss each anemia one by one in the later videos. Thank you for watching. Subscribe for more.